This is TWIS. This Week in Science, episode number 866, recorded on Wednesday, March 9th, 2022. What is the secret to longevity? I'm Dr. Kiki, and I have no idea how to live longer, but we'll learn all about it and fill your heads with all sorts of science here on This Week in Science. I have naps, nematodes, and new approaches. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer. When it comes to the crossroads of science and geopolitics, scientists have a long tradition of ignoring politics and borders and cultural differences, because whatever conflicts governments engage in, science is founded on collaboration, a mutual interest in knowledge, discovery, the furthering of science for the betterment of all humanity. Nothing should interfere with that. And few things do. One thing that does: bombs dropped on towns full of humans. The senseless brutality of the recent Russian attacks on civilians in Ukraine has galvanized many in the scientific community to cut ties with colleagues, organizations, and projects connected to Russia. MIT has ended its partnership with the university in Moscow, which MIT started. The Alliance of Science Organizations in Germany, which funds research, will no longer collaborate with Russian researchers. There are journals that will no longer publish research from scientists at Russian institutions. While CERN and ITER have yet to expel Russian researchers, institutions across Europe and the US are looking for ways to distance their projects from the Russian state. And to be clear, there are Russian scientists who are speaking out against the war. A letter signed by more than 5,000 scientists, including 85 members of the Russian Academy of Sciences, called the attack an unjustifiable war for geopolitical ambitions. While we should always treat scientific exchanges differently than soccer matches, cultural pageantry, and financial transactions, because science is, again, at its core an effort of collaboration towards global knowledge, it is hard to maintain that spirit of cooperation when bombs are being dropped on towns full of humans, and while other fellow researchers are fighting or fleeing for their lives. But we should, if only to show the rest of the world that working together across borders, across cultures, despite geopolitics, and in some cases in defiance of them, is not only possible, but necessary. For This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are here to talk about all the science that has occurred in the last week. Headlines we are excited about, interested in, stories that just whet our curiosity whistle.
Yeah.
And also the politics and policy and things that science is involved in and is affected by and also affects. So, what did I bring for the show today? I have got stories about aging, mice, and marmots, bird learning, and wormy decisions. So much fun. What did you bring, Justin?
I've got hybrid mammoth rats, 10 armed octopuses, Egyptian Bigfoot, and intrepid mishaps.
Intrepid. Oh no. Blair, what is in the animal corner?
God, what is this? Ripley's Believe It or Not. I have shark naps, and then I also have a whole bunch of animal sounds.
A whole bunch. We love animal sounds.
Fun.
Isn't that like a Beach Boys album or something?
Pet sounds.
Yeah.
Yeah. In this case, I have one domestic case and one wild case we'll talk about. They're not all
Awesome.
pets.
Looking forward to it. And as you're looking forward, if you are not yet subscribed to this show, remember that you can find us all places podcasts are found. Look for This Week in Science. We broadcast live weekly on YouTube, Facebook, and Twitch on Wednesdays at 8 p.m. Pacific time. You can find us as TWIS Science on Twitch, Twitter, and Instagram. And if you can't remember all this stuff, head over to our website, twis.org, where you can find information about the show and the stories that we cover after the podcast is published. And now, let's dive into some headlines. Science headlines for this week. There's a few kind of big stories that can be boiled down to headlines, but might be worth a little bit of discussion. Not too much, but similar to your disclaimer, Justin, CERN has not kicked out ongoing Russian investigators involved in science that is ongoing at the Large Hadron Collider. However, they have blocked future interaction and collaboration with Russians and have also removed Russia's observer status. So, like the United States, Russia is not a part of the European Union that is responsible for CERN and the Large Hadron Collider. And so we have observer status. It's been allowed to us. We also put money in, but more so, we're just kind of, they like us, and so they let us play along. But in this case, they have said that Russia doesn't get to play anymore unless things change. And then also on the science newsy policy front, the congressional budget came out today. It just has to be finalized and signed by Biden to really make it to the front, so that finally the fiscal year 2022 will have the money that is going to be dished out for 2022 around the United States. NASA won the congressional budget allotments. Got almost all the money that they asked for. And this is unusual. And the reason I bring it up, it's unusual because normally NASA doesn't get anywhere near what it asks for. So it's very exciting that this year NASA has actually done quite well in comparison to other organizations. National Institute of Health has also received $1 billion. There's a graph that I'm showing on screen right now that doesn't represent this $1 billion that has been allotted to NIH, because it's not really given to NIH. It's been given to the new ARPA movement, which has been pushed forward, and in part by one of our past correspondents, Michael Stebbins. He was involved in part of getting this to happen in Washington, D.C., and moving it to the forefront. But finally, we have money going toward an organization that will be similar to past green technology investments and other governmental investments that spur the creation of new technologies, new research, new ideas. It's more exploratory funding to try and solve big problems, like this is health related. So cancer, diabetes, Alzheimer's disease, some of the big problems that are facing United States citizens currently. So that's very exciting. They did ask for $6 billion for ARPA-H, but they got $1 billion to get it going. So
yeah, you got to aim high. You got to ask for a little more than you need.
Yeah, that's definitely what they do. In this particular fiscal year, basically, a lot of these organizations, governmental organizations, have asked for lots of money, and they are not really gonna be getting what they asked for. But science spending did increase in proportion to other spending across the budget. So at least it kind of increased at the rate of inflation, which, I mean, that might be a bad thing to say right now because inflation's kind of going out of control. But
well, I was going to ask
rate.
What this is it went up in relation to what? Because obviously science budgets were slashed pretty heavily for 4 years. So is this up from last year or is this up from 2 years ago?
Yeah, and so the thing is, the science budgets weren't slashed for the last 4 years. They just weren't given very much, and this is giving a little more, but not much more.
Okay.
NASA, though, they got a lot of money. The congressmen love the SLS. They love this big rocket that we're going to be sending to the moon and to Mars. They love it. And so a big chunk of NASA's budget is going to the SLS, basically because it's pork barrel and it's going to be giving a lot of jobs to many Americans across the country because of the fact that things are made in lots of different states. All the parts are all over the place. But yeah, so good news, bad news. It's, at least budgets for science have not been slashed. And I think that's the big plus here, but it's not what people were hoping for. And then the final bit of science headlines that I thought were interesting this last week. The patient who had the pig heart transplant 2 months ago has died. We don't have a cause of death, but
Can I guess?
Death? No, the heart was working fine. Everything was
Okay.
going great. Yes. No, it wasn't hard. We don't know. They did not have a cause of death. He was doing fine. They said he was doing well enough to participate in watching the Super Bowl. He was recovering from this massive invasive surgery and transplantation. However, and things seem to be going well, so we don't really understand exactly what happened. However, he was not a paragon of health before the surgery happened initially.
Yeah,
So
that would usually be the case.
yeah, but it's because he did so well for 2 months. It still does show that this procedure has the potential to help people. And so there wasn't rejection that they could tell. They got a lot of data. So the fact that he went through the surgery, had the transplant, unfortunately has died, but it's still going to be very beneficial for future transplant recipients and this technology moving forward. Yeah. All right. Well, moving on from these headlines. You got some new approaches to things, Justin? You want to look at things from a different way?
Some different perspective. Always worth doing. So we've talked a bit over the years about the pros and cons of bringing back the Mammoth.
Yes. Always a debate on this show.
Before we need to worry about the if we could, should we's, there's a whole issue of how to do it thing that would need to get sorted out in the first place. So this is published in the journal Current Biology. Team of paleogeneticists have turned their attention to extinct Christmas Island rat, Rattus macleary. Their findings provide some interesting insights both into limitations of de-extinction and possible paths forward. So, when sequencing the genome of an extinct species, scientists faced a problem that extinct species, you've got the DNA, but maybe it's a little bit degraded. You don't have the complete, full, multi-pass genome, which we don't have the multi-pass genome of, most living things today, even if we get a sample, right? So there's the problem of they have all these little gaps, all those little bits of missing information. And the idea has been, not a problem. We'll just fill it in with the closest living relative that it has, right? So with the woolly mammoth, you might take an elephant and say, okay, we're missing this section here of the woolly mammoth. We'll just put elephant in there. And you should end up with something pretty much like a woolly mammoth. So
pretty much, kind of, sort of,
pretty much, yeah. So anyway, the Christmas Island rat went extinct about 120 years ago, which is also very interesting because I didn't know there was, that I would have guessed if you told me there was a rat on Christmas Island, which is this island just off the coast of Australia. I would have assumed it had come over with the boats, the first, some of the explorers. But it turns out the explorers killed off the rats through disease somehow. So right back at you for the whole plague thing. So So they went extinct 120 years ago. And now evolutionary geneticist Tom Gilbert at the University of Copenhagen and his colleagues were looking at, hey, could we bring that back? Let's start small. Let's start with this rat. It's a little bit more recent. So not only were they able to obtain almost all of the rodent's genome from a couple of old rats lying around in a museum, but since it diverged not too long ago from other rat species, they blasted it out and they found that its genome that they have is 95% match with the Norwegian brown rat. Very, very similar. So now they have their
So similar.
elephant for their woolly mammoth model, right? So now they have, if we have the missing gaps, now we've got a perfect model to grab it. So the genome matched up against the reference of this living species. Scientists identified parts of the genome that weren't quite matching up or were missing, where the gaps were. And a few key genes were missing that they specifically know what they do, and they're related to olfaction, meaning the resurrected Christmas Island rat would likely be unable to process smells in the way that a Christmas Island rat had evolved to process smells. So in theory, they could use the CRISPR technology and the gene edit the DNA and remove to take out the portions that they need and then slap them in this new thing. But then you might have this Australian island rat with the olfactory genes of a Norwegian rat, and who knows what effects behaviorally that then have on survivability or its social cues. I mean, this is a creature that relies very heavily on a sense of smell. So all these interesting problems potentially come up. So the Norwegian to Christmas rat ratio scenario is particularly a good test, they say, in this because the evolutionary divergence is actually pretty similar to that of elephants and mammoths. So this is a pretty good model. This is quoting Gilbert. It is very, very clear that we are never going to be able to get all the information to create a perfect recovered form of an extinct species. There will always be some kind of a hybrid. So the replicas will never be perfect. He makes this point, though, that's pretty interesting. It's, for some reason, it's the easier approach, but it's the one I had not really heard talked about before. In order to make an ecologically functional mammoth, for example, one might need to edit elephant DNA to make the animal hairy and able to live in the cold. If you're making a weird fuzzy elephant to live in a zoo, it probably doesn't matter if it's missing some behavioral genes, he says. But then that brings up all sorts of ethical questions. So instead of, the starting with that gene and trying to build it up from a blastocyst into one of those, what were the sheep pouches or whatever they were, or have
Lamb
a
bag.
lamb bag. Thank you. or maybe even inseminate it into an elephant and try to have it give birth. What he's talking about is just edit the genes of an elephant to become more mammoth-like. Would be not that he's saying you should do this. Don't try this at home, people. But he's saying this might be a better approach. And if you know that it's not going to behave the same, but it's going to be a zoo creature anyway, if you're not actually trying to rewild it, it wouldn't be necessarily a problem. But if you were trying to rewild it, it could have all sorts of issues. Gilbert plans on actually trying to do this experiment with the rats. But he's going to start with species that are still living. He intends to begin by doing CRISPR edits on a black rat genome to change it to a Norway brown rat before he attempts the resurrection of the Christmas Island rat.
That sounds like a genetic magician. I will turn this rabbit into a hat. But I'm going to turn this black rat into a Norway rat.
Yeah. I'm going to turn this rabbit into a different rabbit.
So
Yes,
you're
exactly.
you're guessing and assuming what needs to happen to an existing species genome to match a species that no longer exists, which is a lot. And on top of that, the missing sections of genome could have other things on it you don't know about. What
Right, where if
about
they're like, "Oh, it's just repeated segments of DNA." And it's like, well, sometimes repeated segments of DNA, they do have a
Mm.
function in like the number of times they're repeated. It affects translation and transcription and the amount of protein that gets created, and the amount of
yeah.
protein definitely has an influence on metabolism, behavior, etc. So, yeah, there's all sorts of aspects of this that I think the
Yeah.
key message is we don't know enough at this point in time. And like we've been saying, the sensational aspect of, hey, let's make a mammoth. Let's use an elephant to make a mammoth. It's not that easy. You can't just take old
Yeah.
DNA, stick it in an elephant,
No.
ta da! It doesn't work that way. And
Well,
that's,
and I
yeah.
also will continue to take issue with the sentence to make an ecologically viable mammoth.
Yeah. Yeah.
I will continue to take issue with that.
You don't think that there is a currently ecologically viable version of a mammoth?
No.
Do you think it would be better to, knowing that we have climate change coming, help the modification of modern elephants so that they do become better at navigating? I mean, they're already good at hot temperatures in lots of places, but
The
I'm
elephants
is
are
here
there
to
fine.
tell
something we can
you.
do to help the elephants of today move
So we don't.
into
Yeah,
different
I'm
environments
here
and
to
help?
tell
Stop
you
killing
the
them.
answer to that, and it is to get rid of the humans because we are
Yeah.
still killing 96 elephants a day for ivory.
Wow.
That has nothing to do with their ecological viability. Why
Yeah.
would you bring another animal onto this planet to be hunted
Yep.
for
Whatever
poaching
it's got.
purposes? Yeah,
Mhm.
it's
So
Yeah.
mm, mm,
so
mm.
Yeah.
so yeah. Oh no. So
It's
I was gonna tell the podcasting audience that while you were describing the story, telling this the bits and pieces of your story, Justin, that if the
mm,
podcasting audience could only see Blair's face as she was listening to you.
part of
so
the problem is humans are not considered part of the ecology, right?
Yeah, that's a
You
huge
talk about
problem.
like there's space for them or there's a good climate for them in a particular place or there's food for them, but you don't think about humans as part of the ecology, which is a huge piece of this.
Yep.
so a jumping off point here that I think you just brought up there, Kiki. Uh, we've been talking about reintroducing and all the difficulties, and now you're pointing out like, but the ecology isn't there, and you can't really, you don't need to help the current elephants in global warming. Thinking ahead, though, what's the next thing? What's the next big animal that's going to go extinct that we could help by adjusting its DNA today?
Polar
What if
bears.
we made exactly? What if we made
What
bald
can
polar
we
bears
do to help
or
polar bears
less
survive?
shaggy polar bears? What if we?
Yeah.
I mean, is it time to start thinking that in that direction?
So the problem with polar bears is that they go fishing for food, and
Yeah.
with the ice melt, there's no platforms for them to hang out. So I think you'd have to give them gills.
Yeah. I think no, maybe what we need to do is create Arctic uh lily pads.
Yes.
So as the Arctic Ocean warms and becomes more amenable to more plant life, and we could just grow
Mhm.
lily pads on the surface, and then the polar bears could get on the giant
Yeah.
lily pads and fish
Giant
from
lily
them.
pads. Yeah.
Yeah, why not? I mean,
Just
anyway,
giant white lily pads improve the albedo of the planet. You know,
yes, yes.
just
There's a whole bunch of things. Anything you change, it's a butterfly effect, right? Anything you change. But oh, okay, we have so much. We could probably spend an entire day talking about just this topic, but
certainly,
that
certainly.
would make us tired, and I might need to take a nap.
I need to take a nap,
Yeah.
like a shark, like a
Well,
little baby shark.
wait,
No,
can you
sharks
talk?
don't nap. That's the thing about sharks is they never sleep. They're always
They just keep
swimming,
Yeah,
swimming,
swimming.
swimming,
Keep on
or they
swimming.
they
die.
just
Isn't
keep
that
Swimming.
the
swimming
thing
or they
I
die.
learned
Just
when I was
swimming.
7?
So
Yeah,
first,
Hannah learned it.
let me answer that question before I even talk about my story. there are some sharks that have musculature over their gills so they can actively pump water over their gills. There are other sharks who do not. And so sharks that hang out on the bottom of the ocean, like horn sharks or swell sharks, they have those musculatures. So you can see them actually physically resting on the ocean bottom, not moving at all, and they can breathe. Other sharks, larger sharks, great whites, they do not have the musculature to push water over their gills, and so they, quote unquote, never stop swimming, or they will drown. Mm-hmm. Kind of true, kind of not. This story is all about whether or not sharks sleep. In the case of a great white or a shark that would need to keep moving, it is still possible for them to sleep unihemispherically. There are lots of animals that do that. If you ever see a flamingo sleeping standing on one leg, they are also engaging muscles as they sleep.
Whales and dolphins do this.
Yes,
right? They sleep with half of their brain is asleep, and they got the other eye open so they can keep remember to go up and breathe and do whatever they got to do.
Yeah, and so that's not going to stop a shark from sleeping. Basically, just a different process. But so the long and short of this story is, yes, sharks sleep. Okay, that's it. I'm done. No, so this is
okay. The next story we have.
Yeah. So this is a study from La Trobe University in Australia, looking at sharks in Australia and New Zealand. There has been anecdotal evidence from scuba divers saying that it looks like sharks are sleeping. Sharks are fish. They don't have eyelids. How do you tell if they're sleeping? It's all been very anecdotal, and so there's not really a clear scientific answer. But so in this case, they were able to take some sharks into a lab. They put them in aquaria filled with oxygen sensors and monitored them over a 24-hour period. They monitored the physical activity and the oxygen level in the aquarium to see how quickly they are using oxygen to fuel their metabolism. So all of this together, you can kind of put the picture together. Is this shark sleeping? They metabolized oxygen fastest while actively swimming. Makes sense. And the rate dropped when they stopped swimming, and it dropped even further during behavioral signs of sleep. So this is motionlessness for at least 5 minutes with the body held in a near horizontal position. Really looks like sharks sleep. That's it. So that's great.
So you could have finished it back then.
No.
Yes, but
there's
what
more information?
The kind of follow up to this is, why do sharks sleep? In general, if you want to think about like broad strokes why animals sleep, it is to conserve energy while resting. Your metabolism drops, you have to burn less energy, right? But there are potentially other reasons for sleep. Sleep can help with memory storage. It can help with memory consolidation, can help with removal of toxins, can help with healing. There's lots of things that sleep can do. There's a lot of regenerative abilities of sleep depending on the animal that you're looking at. And so sleep is complex and can serve many functions in an animal system. So there are arguments on both sides of the fence here. There are some researchers who chimed in to this research to say, okay, what else are they doing? What's happening in their brain? There are others that say, they're just conserving energy. Chill out, right? So there's people on both sides of the conversation here. The next step to really figure that out is to look at what's happening in their brain. There has been previous sleep studies on zebrafish. They could look at imaging using fluorescent imaging techniques on their brain activity so that they could see what's happening in the fish. And they found 2 distinct phases of sleep in these fish. And I will also add, since sharks are fish, I'm not exactly sure why people were so surprised that sharks also sleep. But regardless, why
would they sleep?
Yeah. The kind of issue with studying sleep in sharks is that the majority of sharks live in salt water, and as you can imagine, electricity in salt water creates a challenge. So it's a lot harder to figure out what's happening in a shark's brain because you have to deal with a conductive fluid, which is an interesting piece of that puzzle. But the one thing that can be drawn here is that sharks sleep, and what's cool about that is that there really appears to be some extremely deep-seated evolutionary history of sleep. And this is why the sharks are so particularly interesting. Oh my god, is that they're old. Sharks are really, really old.
Right, yeah.
Evolutionarily speaking. They've been around for a very, very, very long time. All vertebrates came from fish, and so sharks have a good kind of potential key into the history of sleep in vertebrates as a whole. So there you go.
Well, follow-up question.
Love it.
Mm-hmm.
Follow-up question. What was the plural of aquariums? Again?
Aquaria.
Aquaria. I
The
did not know
age
this.
of Aquaria.
I knew you were going to do that. I almost... When the moon
is in the seventh house.
Yep. You got it. Anyway,
sorry, not sorry.
When the moon's in the seventh house, it's time for the sharks to nap. Anyway, moving on.
Nap time.
Who's next?
Well, sleep has been around for a very long time, but other things that have been around for a long time as well. Getting older, aging, that's been around for a bit. Yeah. Well, researchers they're really working, trying to figure out how to make aging better and how to make more older people seem younger or actually be younger. And so researchers at the Salk Institute have been working on anti-aging in mice. And previously they had given their little anti-aging cocktail to mice. It consists of some reprogramming molecules, 4 of them, called the Yamanaka factors that are OCT4, SOX2, KELF4, and CMYC. I don't know. Anyway, there's these factors, the Yamanaka factors. And if you give an organism or cells these factors, it resets epigenetic marks in the cells so that they're more like their original patterns. So as you're going around your day, awake, not napping and sleeping, but as you're going around your day, interacting with the universe, your body's like, assault. And epigenetics takes place. Lots of mRNA and other things get in there and they wind up DNA. They put little markers on your DNA. They say, ooh, this thing happened to me. And so my cell needs to remember it for X amount of time. And boop, 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 all the epigenetics start changing. And so when you're living, the epigenetics are going, getting you closer to death. And so what they do is they put in these little reprogramming factors and it takes those epigenetic markers, wipes them clean, resets the slate. And they've shown previously that when they give these factors to mice for, a small period of time, that they do have improvement in muscle tone, in metabolism, in all sorts of factors that are normally looked at to count scores of aging. So in this new study, they wanted to know what happens with this regenerative approach in healthy animals as they get older. And so they did the equivalent, two different groups of animals. One group that was the equivalent of starting the treatment at 50 years old and taking it until they were 70 years old. So in mice, this is 15 months old to 22 months old. And then another group was 12 months to 22 months, which is about 35 to about 70. And then they gave a very short-term treatment to an additional group that was just for a month at about the equivalent of 80 years old in people. And so they wanted to find out, okay, if we give this factor to people, right now it's just mice, but if we give it to mice for a long period of time, what's it going to do? Is there going to be a bad effect? Is it going to cause cancer? What happens if you keep wiping the epigenetics clean? And they found that compared to control animals, there were no blood cell alterations, no neurological changes, and they found no cancer as well. And the signs of aging in the groups that had undergone the longer-term treatments were much better than the group that only had that month-long treatment at the later stage. So starting younger, going longer with the regenerative treatment is like a constant resetting. And it didn't seem in mice to have a negative impact on the health or behavior of the animals.
So that's
No.
awesome.
Right.
That's
Is this
awesome.
someday like you're like, I'm 35, 40 years old, and your doctor's like, oh, yep, there you go. Here are
Someday.
your anti-aging factors.
But this is also the problem. This is the problem with anti-aging things like this. Like, oh yeah, we figured out how to keep you from aging, but it's like investing. Yeah, you have to start when you're 3.
Yeah.
What? Yeah. What you do is, just do this one simple thing every other week, but starting when you're 3. And then you're like, but that's not. Now I missed me. I missed it. Yeah, not for you. This is gonna keep the next generation. And then when you're young, you're like, I don't want to do that. I'm young. What do I care about that? That's something some old person cares about, and I don't care about it because I'm young right now. What do I care about tomorrow, guy? This is the problem. Or it's like a genetic thing. Like, oh, you can make a genetic alteration in an infant or before they're born, and
Yeah.
that'll. But it's too late for everybody who cares about it, who's already aging.
Right. So for the young, it's not something that's necessary, and it probably would not even be recommended because you do want to develop. You do want to age. But there is a certain point where people start going, my skin's getting thinner, my muscle tone's not as great as it used to be, my speed, my dexterity, maybe not as fantastic as they once were. And, maybe that's around 35, 40, 45, 50 years old. And if there were a thing, and yes, Blair, I'm looking at you. I'm also looking at myself. But
not at me, because I think a loss of muscle tone is sexy.
It's great. It's awesome.
I wouldn't want it back.
But, if you're not into taking any kind of drug or treatment that is created from this research, you could always talk to a yellow-bellied marmot.
So what?
And ask them for the drugs?
Ask them for their secret of longevity. Did you know yellow-bellied marmots are surprisingly long-lived for their size?
Look at them go.
Yellow-bellied marmots. They're like giant ground squirrels. And they are just the cutest little things. And I got to know a few yellow-bellied marmots once at the Rocky Mountain Biological Laboratory, where part of this research took place. And so I'm very excited to share this study out of UCLA that also, like as I said, took place at the Rocky Mountain Biological Laboratory, Rumble, in Colorado in the Rocky Mountains, where they looked at these animals that hibernate. So when these animals hibernate, they go deep, and their metabolism is like as close to 0 as you can get for a big rodent. And it's surprising, actually, and the researchers were saying that you pick them up when they're hibernating, and they're just like these floppy sacks of fur. Their bodies, they're not doing anything. It's not working at all. And so they looked into these rodents, and what they determined from the metabolism is that because they're hibernating, they're basically going into this very, very deep sleep for a long period of time during the very cold winter. Everything gets turned off, and so there are no more epigenetic changes to their DNA. And so when they're awake in the spring and the summer, and they're going about their life, epigenetic changes are happening, they're doing their thing, the clock's ticking down, and then they go into hibernation, and it's like, it all stops. It doesn't reset it to 0, but it stops. So what I'm thinking, Blair, is that we could just hibernate for part of the year.
What would I do for money?
maybe like 6 to 8 hours a night.
Oh, I see.
All right, now, Blair, I think I caught you fact checking, and I'm assuming you were fact checking whether or not this was hibernation.
No, actually, I was looking up because I always get confused between marmots, marmots,
not hedgehogs,
groundhogs, and woodchucks, which they're all the same genus, so they're basically the same thing.
They're very close. They're just in different locations.
Yeah, but they are sometimes, those words are used for each other at different times. But yes, I don't know.
Except for doing the tongue twister.
Right. No, that's a good question. So, in order for it to be true hibernation, their body temperature has to drop a certain amount. I
Yep.
want to say this is true hibernation.
They're calling it true hibernation.
Mhm.
They are, yeah.
Because the little rodents often do. The problem is, like with
Torpor is
bears,
shorter period of time usually.
and yeah, and so like for bears, for example, they do winter torpor. They don't do full hibernation. They winter sleep is what my professors used to call it. It's because they're so big in conservation of mass and the what it would take to lower that giant hunk of bear meat a certain amount of like temperature is a lot harder than just lowering a little rodent's body temperature and then bringing it back up. And so I think
Mhm.
for a little, not so little, but still little rodent, it's a lot easier for them to do hibernation. But
Yeah, and so, uh, the way it works for these marmots is that they alternate 2 weeks, a week or 2 of hibernation, this metabolic suppression, and then a day or less than a day, less than 24 hours of increased metabolism that then drops back down again for suppression for another week or 2. And apparently, according to the researchers, for an 11 to 13 pound, 5,000 to 6,000 gram animal, they only burn a single gram of fat a day when they're in their metabolic suppression, which is that's tiny, tiny. I mean, look at any food that you eat, it's gonna have more fat than that most likely, except maybe, 0% fat milk, but
what I want to know is
what do you wanna know?
if a human slept, didn't hibernate, because I don't know if our bodies would even do that, but if we slept for like the equivalent. So I was looking and marmots, it's the equivalent of about 50% of their year, right? Is spent
Mhm.
asleep. And so if we slept 12 hours a day every day, would we look younger? Would we live longer? I don't know.
I want you to know that when I was sleeping, trying to maintain my health when I was pregnant. People talk about the glow of pregnancy. I think it's because you're being very healthy when you're pregnant, even healthier than
Right.
than
You're not
than
drinking, you're not
you
eating a bunch of garbage, you're
no, you're trying to
sleeping.
eat well, you're like, you're
Except for the
sleeping
first 6
well.
weeks where maybe you
Right,
can't eat anything and you're constantly barfing.
right, right.
It's that
Yeah.
twinkle in the eye is actually coming from a need to pee constantly.
It is.
Very good.
That is good.
Yeah. I don't know what would happen to us if we slept that much.
Oh, it's my dream in life to find out.
Yeah, but
Uh,
because it
Love it.
wouldn't be hibernation if we were just sleeping for
Right,
12
just
hours a day
sleeping.
every day. Would you become undernourished and underhydrated, and would you have other problems? Like that's the first thing I think of is
Oh,
if you're just
probably.
awake
Mm.
less, are you going to get liver failure from or kidney failure from like not
You
doing
might
the
get
right thing
bed
as
sores.
there's that too.
but 12 hours a day, I don't think you'll get bed sores. As long as
Yeah.
you're like active enough the rest of the 12, I think it's fine. I think people could, well, just study teenagers.
No, but
That'll
teenagers don't
tell
go to
you.
sleep till like 2 in the morning. So they
But then
still
they,
end
Does
up
it
sleeping
matter?
about 8
but they
hours a
sleep till
day.
2 in the afternoon if given the ability. So,
Yeah.
I don't know. Just depends.
Mm. All right. Justin, we love octopuses on this show. Did you find an octopus?
May have.
You might have.
When what was once an ancient shallow bay 325 million years ago in what some people are now calling Montana, a new kind
Right.
of octopus was apparently found. Interestingly, they weren't sure exactly where it was found. They just kind of was found there. It got donated to a Canadian museum in 1988 and then was immediately ignored for the past 30 years.
It was
Uh,
cataloged, I'm sure. It was
It
put
was
on a shelf.
cataloged, and yeah, but then ignored. But that's actually it's really not unusual.
Yeah.
Canadians largely try to imagine that there's nothing south of their border, and they've gotten pretty good at it over the years. And also, museums sometimes have a lot of things that are being brought in. There's not always time or the right person to study them. So their job is to collect. It's not always to have do the full research immediately. New paper published in Nature Communications by a couple of American paleontologists, Christopher Whalen and Neil Landman, got a chance to look at this and examine it. And they find that this border-crossing cephalopod is an entirely new species. So they named the species, I'm going to mess it up, Calypsi mopodi bideni, after President Joe Biden, who had just been, when they were doing the submitting for publication, had just been, he was just been inaugurated, and the authors wanted to recognize his stated commitment to science. So Bideni is about 12 centimeters long and has 10 tentacles. Has suckers, fins, and a triangular, hard, kind of bony tissue inside its body for support.
Okay, I'm going to stop you right there and ask.
Yeah.
Is it 10 tentacles? Is it 10 arms? Is it 8 arms and 2 tentacles?
They're saying it has 10 arms.
Okay.
I switched it to tentacles.
Okay, because they're different.
They are different. Okay.
So octopus have 8 arms.
Okay.
Squids have 8 arms and 2 tentacles.
Okay. So this is a 10-armed octopus, meaning it's a decampus.
Decapus.
Yeah,
Well,
that's about right.
Actually, it's you're right. No, it is.
Decapus?
It's a squid, also.
Okay.
So
because also in the picture in the drawing, it looks like there are 2 tentacles. There are 2 specialized longer appendages.
Exactly.
So does it have 12 appendages?
So I think those are the 2 that round it to 10.
So then it's just a squid. Because they currently have 8 arms and 2 tentacles.
Yeah, it's a
I have
squid.
to admit, Justin, I saw this story, and this is why I didn't bring it, is I got very confused about why this was special.
Okay, so here's where it is. So,
please help me.
Yeah, so these are octopuses and squids are the most familiar types of cephalopods. There's also cuttlefish, nautilus. Fossil record for squishy cephalopods with the ones that don't have shells is pretty awful because when they die, they don't leave anything behind for the fossil record, so they're gone. Very special environmental conditions. Soft parts of the animal can be preserved in rock, and that is the case in Bear Gulch, the limestone fossil site where this species was found. It's famous for this kind of preservation. Does a really good job. So Whalen and Landman described the important parts of the new species' anatomy and got some clues about its identity. They suggest that Bideni's features make it the oldest member of the group called vampiropods. This is the group of cephalopods that includes modern octopuses and the vampire squid. There's a single surviving species of Vampirothus infernalis, whose name actually means vampire squid from hell.
Yes.
So
it looks like a demented umbrella with fangs.
Yeah, so it's
I love it.
It's an interesting place to put it because that is actually more closely related to octopuses than it is to squids.
Interesting.
As is Bideni, even though it has a very squid-like appearance, as you pointed out. So the vampire squid has primitive features that are very much in common with this new species, such as the 10 limbs and the stiff internal shell thingy, triangular shell thingy. No living octopus has either of these. Until now, it was thought that the vampiropods, octopus relatives, originated in the Triassic period, about 240 million years ago. But this new species from Montana pushes that back 82 million years. So quite
makes them older. Yeah.
Jump back in time. Quite a way back.
So then the issue was with the reporting and the headlines put on this story.
Yes.
Because what I saw all over the internet this week was 10-armed octopus.
Yep.
first ever 10-armed octopus. That's what I saw all over. But really, it's a squid relative. But what's weird about it is that it is evolutionarily closer to octopuses, and therefore changes our evolutionary timeline. That is what is cool about this. That's awesome.
Yes.
Very nice.
Blair, you need to be telling people how to report stories like this.
I wish they would just call me.
Hey, wait a second.
Before they did a cephalopod story.
Whoa. I even
changes our view of octopus evolution
as a 10-armed octopus, because that's the lead. That's the tagline.
But it's
now,
a lie.
It's a squid.
It isn't a lie because it's not a squid.
Right.
It's an ancestor, closer related to the octopus.
But I would argue, based on its
morphology, you're not going to go with. You know what I'm going to say?
No. Yes, based on its morphology and the fact that it's closely related to the vampire squid.
Yep.
I would push back on that and say that it could very well be a squid.
But the vampire squid, as I just pointed out, is closer related to an octopus
but that doesn't mean
than
it's
it
not
is to a squid.
You also don't know what the common ancestor with squids and octopuses look like. Maybe they had 10 appendages, and therefore maybe octopuses are the weird ones.
I think we all can agree on that.
The aliens, yes.
The octopuses are the weird ones. That is absolutely true.
But now we all know about this class of vampiropods and their relation.
And that
neither squid nor octopus.
The president has whatever you want to call it named after him.
A cephalopod.
A cephalopod, yeah.
The cephalopod was found in an ancient dried up bay in Montana. Of all places. Doesn't have any ocean.
Unlikely. Right.
Well, it's also kind of. It's also this is actually there. I left out a part that is kind of also I thought a little bit interesting. That existing living infernal vampire squid thing lives in deep ocean. Like drifts around in total darkness at the bottom of the ocean, and they think this ancient relative was living in the shallows. So
interesting. Yeah.
Its closest living relative, and they have very disparate environments that they're living in. Even though, like you say, morphologically, they have some similar appearance.
Well, lots of cephalopod species that boomed and busted.
And we just don't know. I mean, that's also the amazing thing, or the horrible thing, or the intriguing thing about it, is that because they're just missing from the record, when we do find one, there's all these missing points that we would have to sort of connect. Like, how did this go from here to all of these things? We don't have the little transitionals all the way along like we do with many species.
The soft-bodied ones especially. We don't have them.
So tough.
They're gone. Something we do have, at least on the west coast of the United States, are scrub jays. And scrub jays in California are known as the California scrub jay or the Western scrub jay. California's in Florida and other areas are a Mexican jay. And Mexican jays are more social, group-oriented. The young of the Mexican jay, they love to hang out together and help their parents take care of the young. And it takes a long time for them to kind of go, all right, I'm going to go out on my own now. And they might never. They might just stay in that social living group and just spend their life helping out with the family group. And that is totally fine for the Western jays. Now, scrub jays, screechy birds that I love so much. The Western scrub jay is more independent and loves to hang out on its own, very territorial. They form breeding pairs, but otherwise they kick everybody out. They like their own area, even their own young. As soon as the young are old enough to run off on their own, they go, and they chase their babies out. Get out of here. Go find your own future and life. Anyway, Western scrub jays. I love them very much. I think they're fantastic. Anyway, these researchers from University of Oregon and also University of Washington decided to compare these two groups of scrub jays as to how they learn. To say, okay, well, we figure that birds that are social, like the Mexican jay, will be more likely to learn from demonstrators. So they would mimic a behavior, be more likely to pick it up from others demonstrating. And the unsocial, asocial birds are less likely. So the asocial birds are probably not going to pick up on what the demonstrators are doing, and they're just going to learn by their own trial and error process.
Now, I'm guessing by the setup, maybe they found something totally unexpected.
Yeah, they totally did find something unexpected. They discovered that, these Western scrub jays, they learn just as well as the birds in the group, and they learn from demonstrators. They are more likely to try things out on their own than the Mexican jays, but living in a group didn't give the Mexican jays a big advantage over the Western jays in learning how to remove food from naturalistic containers that they'd made into basically puzzle boxes that contained food. The Mexican jays would help each other out. The Western jays absolutely would not help each other. But both of them learned just fine. And so the question is, scrub jays, Western scrub jays, have been shown to have what's thought of as theory of mind. I worked with these birds. They are really smart. They're capable of assessing another bird's intention. So we know that that Western scrub jays watch other birds who are storing their food. They watch squirrels in the environment that are storing their food. These birds pilfer food stores. They also want to make sure that they're hiding their food from other animals. And so even though they're not social or as social as the Mexican jays, it doesn't mean that they aren't paying attention.
Yeah, I have an anecdotal for this, and Blair, you're gonna hate it for all the right reasons. Down in Santa Cruz, a friend of mine, not me, somebody else, started feeding a scrub jay that would come and fly up to his porch. And he started by laying out a couple, to the point where the bird would land in his hand and just take peanuts from his hand and fly out. And, he had this one that would come and visit and show up. And then, I was there checking it out. And I think I came back, visited a couple weeks later. And I was like, oh, has your bird been around? Where's your blue jay? Is he visiting? He's like, no, I had to stop. I had to stop. Because the power line or whatever outside behind the house, he said there was like 50 of them. They were all there.
They're all waiting.
Every time I went out.
Well, he also maybe thought he was teaching the same one, but he was teaching a different one every day.
But when you were telling me that story, I was like, yeah, they might not hang out in social groups, but they watch each other. They're paying attention to everything going on in their environment, even if they're not hanging out together.
Yeah, and I was thinking there might be a different reason than the Mexican jays hang out together, like predators, specific predators in their ecosystem, that there's power in numbers. Yeah. There could be other reasons that they're hanging out in larger groups. It could be an issue with taking care of their little chicks. They might need more help or more protection from predators. There could be other things besides just foraging that cause them to do that.
Right. And one of the questions now, though, is, what was the original social form of these animals? Were these birds originally the original animal that the jays descended from? Were they social and gregarious? And is it the Western jays that have lost that because of their environment, or is it the other way around? Yeah. And so what is the ancestral trait? And if sociality is the ancestral trait, then maybe the Western scrub jay has lost the social part of it, but not the cognitive aspect that goes along with sociality.
I don't think so.
Interesting. Nope. You're like, nah.
No, I'm thinking about it, and I'm just thinking about the fact that, like, bird evolutionary history, in context of other vertebrates, I feel like is still... Modern bird evolutionary history is still kind of new compared to some other evolutionary timescales you could look at. And there's such a wide variety of social dynamics in birds. But I would say large social groups is not dominant.
Well, I don't think that's
wait a
true.
second. What
It
about the
depends
old
on how you
adage,
think of
birds
social.
of a feather?
Right.
Yeah.
So that's part of it too, right? If you want to think about just the proximity, sure. But if you want to think about a cooperative,
cooperation
group,
is the big one. Mm-hmm.
That's not common. So
no, it's not. Territoriality is more common,
right? And so, and that's exactly what I was thinking about. And if you want to think about birds as a group as a whole, I would say it's way more likely that jays in the corvids, jays, they all kind of look at each other and learn from each other, but they're not in these large cooperative...
Yeah, but could we be misinterpreting that? Could we be misinterpreting that? And instead of it being territorial, like, this is my land, that's yours, whatever, maybe they're like, this is my side of the couch. That's your, like, they're still, you know what I mean? Like, they're part of that family. They just need their own space within the family.
And I'm going to say, as somebody who had a scrub jay as a pet for a while, or as a house companion that I let onto my couch.
Oh, wow.
He was actually very social. And I say they were very, very social. The scrub jay really liked having interaction. It enjoyed, like, being on the couch, and it would come over and it would mess with me, but it would come over and pull my paper and peck at things that I was doing. It was like having a cat that was a bird.
Yeah.
It was very interesting.
I would totally agree with that for crows too. I feel like crows are very, like, cat-like. Like, hey, pay attention to me.
Intelligence.
You're not paying attention to me. Oh, you're paying attention to me? No, I'm good. I'm going to go over there. Yeah.
But we are so glad that you are paying attention to us. Thank you for joining This Week in Science for another episode full of science. If you are enjoying the show, please share it with a friend. All right, let's come on back with a part of the show that we love to fill with animals. It's Blair's Animal Corner.
With Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
I want to talk about animal sounds.
Woot woot.
Yeah. So I want to start with a story about pig grunts.
Really?
Oink, oink, oink.
Okay.
This is a study from the University of Copenhagen, and this is looking at, they were the lead research, but it also came out of ETH Zurich and the France's National Research Institute for Agriculture, Food, and the Environment. And this international group wanted to look at pig grunts, squeals, snorts, and wanted to figure out what they meant. They used more than 7,000 audio recordings of pigs. They designed an algorithm that could decode whether the individual pig was experiencing a positive emotion, happy or excited, a negative one, scared or stressed, or somewhere in between. They used commercial pigs, as you might imagine, from birth until death, so for their whole lifespan. And they were able to use those recordings and that algorithm to code these things and come up with a sort of an understanding of pig expression based on the sounds that they make. They were able to demonstrate that animal sounds provide insight into their emotions, no surprise there. But what's interesting is the algorithm was able to decode and understand the emotions of pigs, which of course has a huge application in the welfare of livestock. So you could potentially have an AI, or you could train individuals to recognize sounds, to recognize the well-being of the animals on a farm. And based on that, you could make sure that their lives are good up until the end. And also, I would say, I'm going to throw in there, maybe you could improve the slaughter process so it's not so scary. Because, as an example, they recorded natural kind of impacts on these pigs' lives. So they recorded positive and negative situations. Positives included suckling from their mothers or when they're united with a family member after they were temporarily separated. Negative situations included separation, fights between piglets, castration, and you guessed it, slaughter. So they know what's going on. There's panic, there's smells, there's all sorts of things going on, so that they definitely know what's happening to them, and it's one of the negative emotions. So if you can find a way to make it less negative, that might be nice, right?
Earplugs.
Sure.
I think they should give earplugs to all of the workers and the pigs.
Right. I don't know. I don't have an answer to that. I don't know how to make this better, but if you can recognize that it is a negative experience,
lab-grown meat.
Lab-grown meat would be helpful. If you could find some way to anesthetize them cheaply and where it doesn't impact the meat at all, then they could just be asleep. That would be nice. I don't know. There's ideas. But there are people who actually have to do this for a living and experiment with procedures for the management of livestock. So I'm going to leave them to that. But what I can tell you is that in the second phase of the experiment, they also had experimental stables. They created mock scenarios for the pigs to evoke nuanced emotions in the middle of the spectrum. They know about the positive and the negative. They want to know about the stuff in the middle. This included having toys or food in a corresponding area, and then going into another area where there was no stimuli at all. Oh. Then they also placed new and unfamiliar objects in an arena, recorded them while they were interacting with it. And then on that, during that time, the pigs' calls, behavior, and heart rates were monitored and recorded. And so through analyzing these over 7,000 recordings, they found a pattern. They were able to discern positive and negative emotions. And they also collected, they saw specific trends. So more high frequency calls like screams and squeals were in negative situations. Sounds right. At the same time, low frequency calls, barks and grunts occurred in both positive or negative situations. So barks and grunts, harder to tell. When they look closer, they found that in positive situations, the calls are shorter with minor fluctuations in amplitude, like short and consistent. Grunts, more specifically, would be high and go lower in frequency in positive situations. So they could see these kind of specific patterns related to positive and negative. So you could train the algorithm, but I do think you could also train a human in this as well, which would be helpful. And so they
just from the visualization of the data, it seems so so discrete that these sounds are definitely their own populations of sounds, and that they don't really overlap very much. And so you're right. I think they could be. You could. I would imagine a lot of people who work with pigs, hogs, that they have a sense of what the sounds are already.
Yeah, and talk to any pet parent. They can tell you the difference between an excited squeal and a pained squeal out of their cat or dog or rat or guinea pig or whatever it may be. You can tell. And it's because there's a difference. and you learn them. The algorithm was able to classify 92% of the calls with the correct emotion. So very, very effective algorithm.
Yeah.
So next, they want to develop the algorithm into an app that farmers could use. So if they're not going to use, if they can't use their own ears, the app could discern for them how the pigs are feeling. But with enough data to train the algorithm, they actually think that the algorithm could be adapted to better understand emotions of other mammals, with machine learning, totally possible. I could see that happening. So you feed it enough data after it's kind of the pig as the basis study, they could probably figure it out.
Yeah, and suddenly we understand at least the general emotional state of animals. And like you said, that can influence the way that we treat them and the way that we work with them. Kaivago in the chat room says maybe someone should get those talking buttons for pigs
Yes.
to see if they use them to communicate or if they can learn to use them. And
They
I mean,
certainly could. They're very smart.
I would think so. Pigs are at least on par with dogs
Yeah.
and children, in terms of intelligence. So, at least from what I've heard, so what I've read.
No, pigs are very smart. I bet they could do that. So that all works though if it's an animal that can make sounds.
Right.
You know what animal has not been studied for the sounds that they make until today?
What?
Snail. Is it snails?
It's fish.
Fish.
What?
Yeah, we're back to fish again. Sharks don't nap and fish don't talk. Nope, we're breaking down barriers left and right this week. So
I thought we've talked about fish making noises before, haven't we?
We have. We certainly have.
Yeah.
And the thing is though, there's not a lot of science on it. There's a doctoral candidate, Audrey Luby from the University of Florida, who decided to make a website, fishsounds.net. And she did this with an international team of researchers as the first online interactive fish sound repository of its kind. She argues that fish sounds are as important for understanding fish as bird sounds are for studying birds. And I love that she calls them bird sounds. Feels like a little bit of a dig.
It does a little bit. I agree there. I just said, bird sounds.
All right, bird song, we get it. But yeah, so fish sounds, they're a lot more varied and dynamic than you ever could have imagined. And so fishsounds.net has this repository. They hope in the future to be able to have it open for people to publish their own fish sounds. Right now it's this closed group of researchers, but they would like people to be able to add their own fish sounds in the future. And you can go, it's free. You can go listen to fish sounds. I was doing it for quite a while before the show today. And you can learn all about the different fish and the sounds that they make and see visualizations. It's very cool. Yes, Kiki, I see you're muted.
Yeah, how many people have ever tried recording the sounds their fish make? I mean, we go and we tap on the side of the tank occasionally, right? But
so wait a second.
Get a tin can, stick it in the water, record the sounds. What?
I don't recall fish having ears.
They have the same structures that we do. They have inner ear bones. They have all of the same kind of mechanisms. They don't have an ear hole, but they can totally hear and respond to vibrations. Like snakes don't have ear holes.
Why would you have an ear hole in the water? I mean, I guess you could. They've got the gills with the gill slits.
But
the water comes in and out. But
maybe I
miss.
Maybe I have a big missing gap in my evolutionary history. But I thought the inner ear bone was like a separated part of fish jaw at some point. That wasn't there something.
So I know what you're talking about. I do believe it's like a lamprey jaw. It's like it's a jawless fish. Structure that looks like we'll talk about it in the after show, but I do believe it is like a pre-fish is what you're talking about.
Okay.
It's not actually fish as we know
so
them.
Fish have ears.
Yeah, they have structures for hearing and responding to vibrations and sounds. Yeah.
Like up in their head, like behind the jaw somewhere.
Yeah.
Wow.
So Blair, what were some fish sounds that you wanted
Thank you.
everyone to
Yes.
hear?
So this brings me to my favorite part of the story, which is that there is not yet a standard system for naming fish sounds. So we were just talking about pigs, and we talked about squeals, and we talked about grunts and stuff like that. There's no standard system for naming fish sounds. So this project has had to make them up.
I mean, we have a lot of sound names
Yes.
already, so it's not completely making things up.
Right. But if you start to holistically kind of look at this database of fish sounds, there are types of sounds that keep coming up. And my favorites are there are boops, boop. There are croaks. There are boat whistles. There are grunt thumps. There are unks. Very good. But so some of my favorites were from toadfish, and I think most of the ones I sent you, Kiki, are from toadfish.
Okay.
But let's see.
Yes, so this is a Halobatrachus didactylus, Lusitanian toadfish. It's a croak.
Very good. That sounds like a frog, right? That does not sound like a fish.
No.
Yeah,
I wouldn't.
very
I wouldn't.
froggy.
Here we have a Lusitanian toadfish with a boat whistle.
Oh, wow.
I do like that. The boat, but I wouldn't have thought of it as a boat whistle. It doesn't sound like a whistle.
Sounds a little like tugboaty. Whatever that thing is. The tug.
Doot, doot.
So
I'm not seeing the whistliness.
Okay, Blair, should we reveal? Okay, these are just toot noises that me and Blair recorded earlier and decided to play for you.
No. These are not toots. They are fish.
So, okay.
Here's another one. I can share a picture of the drawing.
But how amplified are these from the recordings?
Toadfish.
Like, do other fish know that fish are making these sounds? Is their hearing sensitive? Are they hearing this?
Yes, they have ears.
Yeah.
But is it loud enough? Like, I don't know, like...
So remember, if you've ever made sounds underwater, like in the pool, that water is extremely... sound transmits very easily in water.
Yeah.
And so it's actually that's one of my favorites. It's actually very it's easier in some ways to hear sounds in water than in the air. There's less resistance, I guess. But so and to answer your question about fish ears, they have their own inner ear and cranial cavity. They have no external or middle ear. They have otoliths in there, which help them to hear, which is like a very, very dense section. So it's denser than the water around it. And you're right, there are gill structures that have migrated into our inner ear.
Gill structures, okay.
But they still have these nerve centers and structures for listening that are analogous to our ear. Right. So you got some of it there, but it's more about the bones. So the bones and the gills moved up into the inner ear, but the organ structures and stuff are analogous. So anyway, they have ears. But yeah, so I could listen to these all day.
You're redlining a little, Kiki.
Well, anyway, go to fishsounds.net and just poke around. Find some unks, find some boops. Have a grand old time. But the point of the story, besides just telling you all to go to fishsounds.net, is that there are active sounds that some fish make, which is mostly what we listen to today. I chose the active sounds because they're more fun. That's when some fish, like toadfish, have evolved organs or other structures that produce sounds. So it's like they're making grunts or unks or boops on their own. But other fish
Which I would not have named these sounds.
Yeah, I don't know.
The names that they have chosen. I don't see it, but I'm not a fish person, so
I think part of the reason
I know bird people's sounds also don't always match up to what it sounds like.
And part of it is that to us, these all kind of sound the same. But to someone trying to categorize these sounds, you have to differentiate them. So
you do.
Okay,
sure.
That was more of a boop, and this was more of an unk. Okay, great, got it. But yes, so there are active sounds like the ones that we all listen to. There are also passive sounds: chewing, splashing, snapping, and so those can also convey information. As Justin brought up, sound is an effective way to communicate underwater. It travels faster underwater than it does in air, and there are lots of spaces where there are lots of fish and low visibility. So sound would actually be a great way to communicate if there is not a good way to visually communicate what's going on. They may communicate about territory, predators, food, reproduction, any number of things. So it makes sense that fish would communicate with sounds. And this is just something that is not really looked at too much now. So the hope is that since this database exists, it will continue to grow. People use their hydrophones underwater microphones to gather more fish sounds. They will be able to, based on the knowledge of fish sounds, be able to use hydrophones, hear what's going on, and draw conclusions about what's happening live where they are. There are under 1,000 fish species that have been recorded making active sounds. That has to be an underestimate. So that is the other really big hope from this, is that drawing attention to the sounds that fish makes, that more people will go out and research this, collect this, catalog this, and be able to look at it as a way to study fish communication. Because I think also fish often get overlooked. People think that they're not that smart, they're not that complicated, there's not much going on in their brain. That whole idea that goldfish have like an hour memory, we know, is debunked,
it's just
right?
Not true, yeah.
So this is kind of more of that, wanting to look at how complex communication might be happening with these fish with sound, which is so cool. Not to mention that, as I said before, we all came from fish. And so is there some sort of auditory communication that evolutionarily is rooted all the way back to our fishy origins? Burp. Yeah, and that's the answer. Burp. 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 Burp.
Who knows? This is This Week in Science. Thank you so much for joining us for another episode of science-filled fun. We know that you are going to be making lots of fish sounds moving forward. If you're enjoying the show, please head over to TWIS.org, click on the Patreon link, and choose at your level of comfort to support us, to keep this show going week after week. We do appreciate your support. We really can't do it without you. All right, coming back with more This Week in Science, Justin, tell me a story.
Okay, let me see. One of the world's most storied shipwrecks, that of intrepid Antarctic explorer Ernest Shackleton, the ship Endurance, has been discovered at the bottom of the sea off the coast of Antarctica. Endurance was discovered at a depth of 3 kilometers, which, if you're not familiar with kilometers, is about 3,000 meters under the water. If you don't remember the Endurance, and if you don't remember Shackleton, why would you? Because it was over 100 years ago. It was once upon a time there was a race to explore the South Pole. Nobody'd been there. It was uncharted territory. Shackleton, the intrepid Shackleton, and his crew got within 100 miles of the pole in one expedition, but were forced to turn back, almost dying in the attempt. Things got so bad they had to eat their horses. And actually, I think they got dysentery and sick from eating their horses, which then that was really what almost finished them off. But it was the closest anyone had ever gotten to the South Pole, and at the time, so they also managed to climb a volcano. This is another kind of thing that the explorer people would do back in those days: is go climb something that hadn't been climbed before, put a flag on the top of it, do the hero pose, and then come back down and tell people about it, because they usually didn't have cameras around all the time. A few years later, after Shackleton got close, the pole would be reached twice by other people. Since he had nothing better to do, Shackleton decided to walk across Antarctica, crossing it at the pole, a feat yet never accomplished, and so worthy challenge for an intrepid explorer such as himself. But the expedition didn't get too far. It got all the way to Antarctica, but then it got trapped in sea ice. The boat, the Endurance. So, since I started this story by telling you that they found Shackleton's ship at the bottom of the sea, you probably know what happened next, or think you do. The intrepid Shackleton and his resigned to their fate crew survived on the boat, trapped in the ice, for nearly a year. The hull slowly getting crushed. Water slowly working its way inside the boat over about 10 months, until finally they had to leave the sinking ship. And so they camped on the ice for a few months, on the ice floes, as the ship continued to go further into the water. That plan seemed to be unsustainable for the long term, so they took the small escape boats from the sunken ship, and they made their way across 1,400 kilometers of stormy Antarctic Sea in basically these little dinghy boats. First landing on an island that turned out just to be covered in ice, not where they wanted to be. They kept going, finally made it back to civilization. They survived. They lived. They're not on that boat that just got discovered at the bottom of the Antarctic. This current expedition, which had a modern-day icebreaker ship, was organized by the Falklands Maritime Heritage Trust, and they used sonar as well as underwater drones to find and film the shipwreck. They found the Endurance also only 6 kilometers away from where the coordinates where the crew had reported their ship having gone down. So it was very, very close to where the ship initially sunk. The underwater drones got some really nice images. I don't know if you've got the link there. Oh, yeah, you got it. If you look at that image, one of the fascinating things, that thing looks intact. You can see there's rope rigging that is up on the deck with the gear. You can see the name Endurance blazing across the front. There's the old piratey looking ship wheel run that
It's
rudder.
because it's so cold.
It's so cold.
It's all preserved.
It sunk straight down. There's nothing. There's a little bit of barnacle-y life forms and such starting to make it its home, but this boat is almost immaculately preserved under there. It's even sitting upright. It's not like on its side. It's not got big holes in it.
It's like the ice just kind of went, oh, we're just going to put you down.
Just
there.
need to just settle down to sleep.
I don't want you up here anymore. We're just going to put you down there.
Yeah. So pretty much upright intact, rigging gear on the deck. Looks like it was sunk yesterday. Under international law, the wreck is now a protected historical site.
Oh.
So the explorers that located it filmed it and scanned it, but did not touch the ship. Are not going to be retrieving any artifacts. Not taking anything to the surface. It will stay there. A international historic wreck.
I'm sure they will send underwater archaeologists down to take a look at it and to
Maybe.
get
They can't touch
inside and
anything.
study it? I don't
No,
know.
no, they're not supposed to go inside. They're not supposed to touch it. They got to just leave it where it is. So the crew, I guess, I don't know if this was just a side thing. So they were doing some climactic research there as well.
Yeah.
They were, doing ice flow stuff, everything, things like this. But I guess they just knew the boat was down there somewhere, and so they got a sonar hit around the area where it was recorded to go down, and they were the quotes are pretty much something along the lines of like, "We knew it. We found the boat because there's nothing else here. We're like the
There
fourth
wouldn't be
boat.
anything else there.
No, we're like the fourth boat
Yeah.
ever to even be in this little bay area.
Yeah.
Like nobody comes here. There's no good reason to be in this place other than some climate research and looking for this particular boat.
And as we've heard, there's no good reason for people to be going there even now.
No.
We're bad for Antarctica.
And speaking of people with no good reason, occasionally there is something on the planet Earth that, through whatever chance of evolution, through some failure to adapt to new environments, life just stops finding a way. Many extinct creatures are extinct for a reason, because they just weren't good at surviving. They just didn't want it. The largest ape species, the 10-foot tall central casting equivalent of Bigfoot, Gigantopithecus, died out 100,000 years ago. 10-foot tall ape. You would think, hey, you're the 10-foot tall ape in the room wherever you go. The world is your oyster. But climate change killed off its preferred forest fruit that it liked to eat. So it just decided to stop existing. It just gave up. Blair might even suggest maybe even a current life form that seems bent on its own eventual demise.
Whatever could you mean?
Pandas, maybe? That's the one that came to mind. Societies can also be like this, though. Societies can mirror this. We have whole civilizations that have come and gone on the planet due to their inability to adapt to a changing environment. You got the Anasazi, classic Maya, Harappa, Hittites, ancient Egyptian cultures. And now, maybe we're gonna see it happen again to the Floridians.
All right, you got to explain this one, Justin.
Like a bunch of lazy pandas, Floridians can't be convinced to help themselves. Despite years of warnings from top scientists around the world, Florida's government seems not just uninterested in cutting greenhouse emissions. The last time Florida legislature addressed greenhouse gas emissions, it was with a law that blocked cities from cutting emissions effectively. Florida passed a law, it was actually crafted by natural gas companies, that prevented cities from banning the use of natural gas. Natural gas provides about 70% of the state's fuel at power plants and is also used in homes for cooking, heating, everything else. It's made of methane gas, which heats the atmosphere much more than carbon dioxide does. So their new law undercut individual cities' ability to meet goals that they had set for themselves to get to net zero carbon emissions by 2050. Because instead of doing electric or different types of cooking, you couldn't ban natural gas from your town. So they have been spending hundreds of millions of dollars attempting to raise roads and flood-proof buildings because things are already happening. The flooding, the water rise is already happening, to where they're spending hundreds of millions of dollars attempting to adapt to a changing environment, but not going to address the actual problem behind it.
Well, Justin, it's already happening. Why do anything if it's already done?
Yeah. So, and now Florida ponds.
Not kidding.
Florida ponds are getting into the spirit. Turns out Florida, which has had some rapid residential growth over the years, has some 76,000 man-made storm ponds, and these ponds emit more carbon via gas, apparently, than they store in their muck. They're negative carbon sinks. So older waterways are pretty okay at carbon sinking, but the newer man-made ponds lack the biological infrastructure to do any sort of sequestration, and so they
Right.
end up actually giving off more gas than they retain. So,
Huh.
uh, like
So
the
they're doing the roads and they're doing all this stuff, but they're creating these urban stormwater ponds, but they're
mhm.
not making them right.
They're not and given enough, given a generation or two, maybe they'll be in equilibrium. But right now, these they got tens of thousands that are actually doing the opposite of what you would otherwise.
Well,
This is
but
a
isn't that
study
because
by
they were
University of
made
Florida.
to prevent flooding, not to hold
They weren't
carbon.
made for sequestration.
Right.
Yeah,
Right.
but they're also contributing to their own demise there. So, like the Gigantopithecus, like the dodo
Oh.
bird, like the future pandas, like the Egyptians, like everybody else. Florida, you will be missed on maps. One day it'll just be water there.
Well, hopefully, research like this will get integrated into new stormwater pond designs so that they're not just made to hold stormwater, but actually made to fit within ecosystems and sequester carbon dioxide. Because wouldn't that be nice? Let's design for multiple uses, not just one. Yeah, let's do that. I have some stories, not about Florida. Nope, not about pandas. Not about no, I've got stories about little teeny tiny nematodes.
Nemes.
Nematodes, worms. Yes, little worms that only have about 302 neurons in their
Who needs them
quote
Yeah,
unquote
it's a
brain.
lot.
Right.
in
So
their nerve clump?
we have tens of billions of neurons in our brain, and we think it makes us so special. We're like, oh, we're not fish anymore. Look at us go. We stay awake and we don't take naps. But we do. Some of us really like to take naps. But we also think that all these neurons make us so great at making decisions and doing things. But along come little nematodes, parasitic nematodes that like to um, eat C. elegans, which is another kind of nematode. So these researchers at the Salk Institute were looking at predatory worms called Pristioncus pacificus. And these predatory worms like to eat little bacteria patches, and they also like to eat C. elegans larvae, and they bite. They're biting worms because they're predatory. And in the biting that they do, it turns out they don't just have one strategy of biting, and it's not just for eating, not just for predating. Oh no, these wonderful worms, these parasitic worms, they also like to bite the adult C. elegans because it just so happens that the adult C. elegans like to eat the bacteria just like the P. pacificus like to eat. And so these researchers at the Salk Institute decided to record the activity and look into the activity of these worms. And they saw that it had very complex behavior and that they enjoyed biting and eating the larva and were predatory in their actions toward the larva. But then when it came to attacking the adults, they were much more likely to have a different kind of biting behavior that was just too territorial to scare the big adult C. elegans away. And then they found that when they used a molecular trigger, they were able to switch the behavior of the worm from territorial to predatory. And so there's a very simple behavior that the worms use signals that are released by the bacteria that they eat. They sense the bacteria that they eat and they use that to trigger their behavior. And they also have senses that determine whether or not they're going to eat baby C. elegans or whether or not they're just going to attack the parent. These little tiny worms, very basic signals. It's either attack or get off my lawn.
Attack or attack. That's what it
Attack or
sounds like.
attack. It's just two different kinds of attacking with very different behavioral reasoning.
It's attack to kill versus attack to disarm.
Yes. Yeah. So yes, these worms are. You spent all day listening to fish sounds. I spent all afternoon watching worms attack worms, and I really enjoyed it.
Good. That I gotta tell you, that little lump of nerves, it's up to something.
Little ganglia, right? Little worm ganglia. They are up to something.
The nematodes are great for that. Just like you just have this lump of nerve cells, but it really feels like you have a brain.
It's pretty much a brain. I mean, it does a lot of stuff. Does most of the stuff that allows an organism to survive. And I think that's one of the most interesting points about so much behavior is that there's a lot of it that's automatic and is at the cellular level. And we look at ourselves as these complex behavioral creatures who can plan and think and conceptualize. And yes, but at our heart, when we think, so much is our ability to be conscious of stuff. Like there's a lot going on underneath the hood that would be going on anyway.
Mm-hmm.
So I think, the basics of behavior don't require a lot. And so, what is it? The steps that make us exactly who we are. Are you worm or are you human?
Or are you dancer?
Exactly. I should write the lyrics to their songs. They might actually make more sense. And my last study for the night that I want to share has nothing to do with worms and has everything to do, Blair, with aging.
Yes.
Yes. One more study on aging. And apparently, you have to go to the Amazon's indigenous tribes to age really well without dementia or Alzheimer's. So, researchers did an observational study that was just published in the journal Alzheimer's and Dementia, the Journal of the Alzheimer's Association, in which they looked at several hundred people from the Tsimane tribe and also the Mosetan group, and they looked at the over 60 age group to determine what percentage of them suffered from cognitive impairment, Alzheimer's, or dementia. And they found that these two groups have much lower rate of dementia than anybody else that they've ever looked at on the planet. Only about 1% suffer from dementia. And there are about 11% of people in the United States who suffer from dementia. And so one of the questions is that researchers are trying to parse out, and this study does not do that, but researchers are trying to determine whether or not it's the urban lifestyle that has led to the deterioration of mental state as we age, or are there other factors at play? And as I was reading through this study, I thought it was very, very interesting to look at these different groups of people, and they compared also their results with these Amazonian tribes with a review of 15 studies of indigenous populations in Australia, North America, Guam, and Brazil. And they found that study earlier found a dementia prevalence that ranges from about 0.5% to 20% among indigenous older adults, and with the highest levels being more so in individuals that are closer to urban life. And so that seems to support their hypothesis. But the thing that they don't do that I wish they had been able to do was to figure out whether it has to do with the number of people who have just survived in their environment to the age at which they are included in the study and don't have dementia. The people in the Tsimane tribe are subsistence farmers, and so they live are subsistence livers. They subsist on things from the environment. They're foragers. They don't have malls and stores and places that they can go and, they don't have fast food to go get whatever they want. So is it that they're able to survive better because they don't have a predilection toward mental disability, or what is it that led them to that low number in the first place? And so that the study doesn't answer that really, and it would be nice to know.
Yeah, that's interesting. I was reading an article today about downtime and how we're doing downtime wrong.
That's a clickbait headline. You're doing downtime
It
wrong.
certainly
right?
is, but this is from the Neuro Leadership Institute, and they were talking about how your brain needs actual downtime and how in modern society, time that we designate as downtime is not watching TV, listening to a podcast while you do the dishes. This is not downtime because you're actively engaging your brain in an activity. You're not allowing it to kind of move and flow and all this kind of stuff. And so I'm wondering about that. I'm wondering about a disconnection from the hyper connected lifestyle that we live, the hyper efficient, gotta use every spare moment of our lives to better ourselves or constantly be engaged, if that could have something to do with the health of the brain.
Well, additionally, we know that there are influences from the light that we subject ourselves to constantly. Televisions, LCD screens, artificial light. We're not just dealing with the light of the sun anymore. We're not. And so that light is affecting not just our eyes, but also the clocks in our brains, and blue light versus warm light has different effects because these are different frequencies of light, and the different frequencies of light stimulate our neurons differently. So there's a whole bunch of factors that get involved there, but it's all confounding factors. Is it urban life? Is that what's doing it? Is that what's leading us to have more more mental disease, more mental deterioration as we age? What is it? We don't know. It's so many confounding factors, but very interesting. We should go become part of the Semaine tribe if we want to have lower risk of dementia.
So first of all, I would like to express my opinion that listening to TWIS is perfectly acceptable downtime. And you should continue to listen to the show to relax
Absolutely.
whenever you get the chance. But I would also point out that, I don't think I believe the data behind this the comparative data for the, I was just looking up this global collection of studies. It shows that the United States is 14% dementia, but it's after 71 years. The other places, South America, it's 7.3. But then, it's so dramatic. Europe is 6, and China is 3. I feel like there's a lot of different definitions and levels of diagnosis and everything else going on, because I don't believe that life in Europe and life in America, city or small town, is enough to be 7 times or 4 times or 5 times as much dementia. I think I have a problem with
pollution
the underlying.
Is probably I would believe pollution would be a big factor in that.
Sure, but then China's only 3%.
Right.
In
well,
their
a lot of China
version.
Is rural, but
population densities. Maybe it's just more recent that's become. But the I don't know. I have whenever I have a diagnosis of especially dementia, which is a spectrum-y kind of
right.
thing.
It's not a specific disease. It's a spectrum of. It's a cluster of symptoms.
Yes. What I would say though is that it might be more obvious in a highly urbanized city area, someplace where you got to take 3 subways to get over to the grandkid's house, and then you're going to jump onto the F train because it's the nighttime or whatever's going on. Maybe those problems manifest easier with a more complicated set of obstacles in front of you or tasks in front of you on a daily basis. But I don't know. I have a hard time. Anyway,
I think I mean the point I was trying to make with the complexity of whether or not people survived to be included in the study is that potentially it's not just the urban environment that could whatever factors, pollution or distraction, and whatever it is that circadian rhythms, whatever, that lead to dementia and Alzheimer's disease. Possibly, I mean, it could be that we have a better medical system within urban centers to allow people to survive longer. We have more, even though the United States has, we talk about how bad medical care, the healthcare system is all the time. At the same time, it is better than in the jungles of the Amazon. So when somebody, steps on a nail here in the city, we can go get a tetanus shot. It's a lot harder to do when you're in the jungles of the Amazon. We have vaccines that are more available.
There's a lot less nails, though, to be fair.
No, I'm making examples, but the likelihood that, an injury or an illness is going to fell you at a young age is higher in a less urban area, like or a more remote area, than it would be in a city or in a place that has a good healthcare system. And so COVID-19, for example, we're now seeing that, and this is one example, and there are other viruses that potentially lead to lifelong illnesses as well, but we're finding that COVID-19 leads to shrinkage of your brain. It leads to potentially long COVID in about 30% of people, and that you have the potential for things like multiple sclerosis or other autoimmune diseases to pop up after you have a viral infection. So say in the Amazon, you get COVID-19 and you die. Here in the United States, you get a ventilator or you get the medication, you survive, but then you have a virally induced illness that later predisposes you to Alzheimer's or dementia. So perhaps it is a factor that, more survivability. I mean, so many confounding factors. There are so many confounding factors, but like earlier, Blair, you were saying about the way that the octopus vampire squid story was covered. It wasn't, you didn't see what was going on there. And that's like with this study, this Amazonian reduction in dementia that I've been seeing headlines all over the place. It's like, oh, it's this amazing special group. And that is not what the study says at all. It's an observational study that says we found this number.
Yeah.
It doesn't explain why. It doesn't explain how. and they actually, and they go through this whole thing, and there's like one sentence that says, yeah, there could be these confounding factors about, related to who survived. So sampling bias of their age group. Yeah. Anyway, the headlines. Don't just read the headlines because they don't tell the whole story. And sometimes even the stories don't tell the whole story. It's important. And yes, Amazon, Blair, don't book those airline tickets to the Amazon.
No, don't worry, I won't.
Not just yet. We have made it to the end of the show. Isn't that amazing? Isn't that amazing? We made it. We're here at the end of the show, and I would like to say thanks so much to Clarissa M. Chains for listening to and enjoying the TWIS podcast enough to list us in their top 10 podcasts list, and wrote a blog post about it. And so we'll link to the blog post in our show notes. Very nice group of podcasts to be included in, and I appreciate being listed. And so thank you very much for doing that, Clarissa. Clarissa also, and this is for a later show, not for tonight, so we have homework. Okay. Clarissa wants to know our thoughts on the USAID's Deep VZN program. And this is a program to identify viral variants And so there are aspects of the openness and transparency of this program that we need to dig in on it. So we have homework to do, research to do, so that we can discuss the USAID's Deep VZN program on a later show. But in the meantime, it's there. And Clarissa, thank you for the discussion topic, and thank you for writing in. Thank you, everybody who writes in and sends ideas for interviews and other things. And if you have questions, ask us questions. We'll see what we can answer. But we are now at the end of the show. So thank you all for listening. Thank you for joining us for another episode. I do want to do shout outs because that's what time it is, isn't it? It is time for shout outs. Shout outs to Fada. Thank you so much for joining us and helping with the show notes and show descriptions and social media. Really appreciate that. Identity 4, thank you for recording the show. Additionally, who else do I have to thank? I need to thank Gord and Arenlor and other people who help to maintain the safe nature of our chat rooms. And thank you to Rachel for editing the show and for your assistance on so many other things. Thank you so much. And to our Patreon sponsors, thank you for your support of TWIS. Thank you to Richard Badge, Kent Northcote, Pierre Velazarb, Ralphie Figueroa, John Ratnaswamy, Carl Kornfeld, Karen Tazi, Woody MS, Chris Wozniak, Dave Bunn, Vegard Shefstad, Hal Snyder, Donathan Stiles, aka Don Stylo, John Lee, Ali Coffin, Maddie Perrin, Garv Sharma, Ragan, Don Munna, Stephen Alberan, Daryl Myshak, Stu Pollock, Andrew Swanson, Fredis 104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles, Jack, Brian Carrington, Matt Bass, Sean and Nina Lamb, John McKee, Greg Riley, Mark Hessenflow, Gene Tellier, Steve Leisman, aka Zima, Ken Hayes, Howard Tan, Christopher Rappin, Dana Pearson, Richard, Brendan Minish, Johnny Gridley, Rami Day, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Bob Coburn, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Philip Shane, Kurt Larson, Craig Landon, Mountain Sloth, Jim Japose, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Disney, David Simmerly, Patrick Pecoraro, Tony Steele, Ulysses Adkins, and Jason Roberts. Thank you for all of your support on Patreon. And if you would like to support us on Patreon, please head over to twist.org and click on the Patreon link on next week's show.
We will be back Wednesday, 8 p.m. Pacific time, broadcasting live from our YouTube and Facebook channels, as well as from twist.org/live.
Hey, do you want to listen to us as a podcast? Maybe while you do the dishes? Just be sure to have some downtime later. Just search for This Week in Science wherever podcasts are found. If you've enjoyed the show, you can get your friends to subscribe as well.
For more downtime on anything you've heard here today, show notes and links to stories will be available on our website, www.twist.org.
You can also contact us directly. Email Kirsten at kirsten@thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twist.org. Just be sure to put TWIS, TWIS, in the subject line, or your email will get caught in some ice. It will slowly get overtaken, and it will sink to the bottom of the Antarctic. We won't read it.
Oh.
You'll never know.
Maybe in a couple hundred years they'll find it. But for now, gone.
And you can also find us on the Twitter, where we are at Twist Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your downtime. If there's a topic you'd like us to cover or address, a suggestion for an interview, a haiku that comes in the night, please let us know.
We'll be back here next week after some quality downtime, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This in science. This week 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 in science.